ऐसे नहीं तो वायर्ड इससे यूज करूँगा। इससे नहीं हो रहा तो वायर्ड वाला मैं तो अपने लैपटॉप पे वायर्ड वाला ही करता हूँ ये ना।
Yeah. Good afternoon, everyone. I am Dr. Nupur Mondal, and I take immense pleasure in introducing the speaker for today's session, Ms. Banisha Fukela, PhD Research Scholar and SRF, Department of Botany, University of Delhi. Ms. Banisha has completed her education from University of Delhi, that is BSc from Acharya Narendra Dev College, MSc, and the ongoing PhD as well. In a very short span of career, she has been awarded prestigious fellowships and awards. Since the beginning of her academic journey, she had keen interest in the world of bioinformatics, as a result of which she has written and published written research article as well as book chapter in highly reputed international platforms. She was also the co-editor of Botanica, a magazine published by University of Delhi. She has attended several seminars and conferences and have even presented posters. And the list continues. We are very sure that Ms. Banisha has an illustrious career to look forward to. Without taking any more time, I would like to hand over the virtual dais to Ms. Banisha to deliver her talk on an introduction to bioinformatics. Ms. Banisha?
Am I audible?
Yeah, you are audible.
Yes, you're audible.
Thank you so much, ma'am, for your nice introduction and giving me an opportunity to interact with the students and for sharing my knowledge with this young youth. So today we will try to definitely understand an altogether different discipline, which is bioinformatics. Now, this field introduced when Human Genome Project was started, and it was realized that the amount of sequencing data that would generate would definitely require a platform which can handle data, and it should be accessible to everyone in the form of that we could mine, retrieve, and analyze. So, experts from different disciplines, for example,
I think, Banisha, I think you are on mute. Please unmute yourself.
Banisha, any problem?
sorry for the interruption. Actually, my slides was not moving.
Oh, no. We saw a few. Now it is fine. You can
Okay.
proceed.
So, I was trying to say, so this bioinformatics field was introduced when initially the Human Genome Project was started, and it was realized that the amount of sequence data that would generate would definitely require a platform which can I should say, which can handle an ample amount of sequencing data. And across the globe, people, researchers, biologists, anyone can access and share this information. So therefore, experts from different field
Banisha, I think you stop sharing and then again start sharing. So this problem would get resolved then. Yes, yes, the next slide came.
Banisha, you can share your problem with us.
Actually, I have a problem in moving the slides.
The arrow keys are not working. If not, then stop sharing once and then again start with the sharing process.
Okay, okay, fine. So, okay, I think we should continue.
Sure. Please go ahead.
So, people from different disciplines came together. For example, there was a need of computational biologist. There was definitely a need of statistician and mathematician, and of course, a biologist who all can integrate their expertise and develop the platform. So, bioinformatics is nothing but just an integration or just a amalgam of these four fields. So, all these experts when come together and develop a platform. So, this field is therefore, and one who is expert from all these four areas, we call that as a bioinformatician. So, uh, now now it was a way back when we had a human genome project sequence. Now at present we have an ample amount of sequencing data. There are 100 or 200 genomes that have already been sequenced for, say, for plants and for animals and for other kingdoms, for example for fungus, for alga, and also for the lower plants. And so, and this field has expanded to an extent that we have specialized databases for all these plants. So, now these all four experts are doing their work simultaneously, and eventually we being a botanist or biologist doesn't always or need not necessarily have information or from all these four fields. For example, being a biologist, I need not to learn all the languages that computational biologists have, or any modeling that mathematicians can do to develop these, or a statistical area. But because all these experts are doing parallelly their work, and they have developed those platforms and those databases for us. So being a biologist, we should know only that how to use those databases, those platforms, so that we can resolve our hypotheses, our questions. So for this, first, I would like to question the students that, because we should know the fundamentals of biology, because what is there in the databases, in the different platforms, one should know the fundamental of a genome, because all we see are different morphologies and they are encoded in the form of DNA. So I would like to ask students, so how do you categorize a gene or define a gene? Because if a student know the properties of a gene or a genome, then it is more easier for them to use those bioinformatic tools or those platforms. So this is a question to students. So if I'm asking you that what is a gene and what are the features of a gene, I want to know from your side. So is there any student who can answer or define up just a gene definition?
The coding part of a
Ma'am, gene
sequence.
is
Okay, the coding part of a sequence is answer from a student. And because if you don't know the properties of a gene, you cannot use those databases and eventually they won't lead you anywhere. And if you know yes, this is a gene, this is the information it has some regions which are specific to it, and therefore we call that gene, a specific genome. Then only we can start thinking about those platforms and can start with the with those tools. And it is a coding, and which part codes for any RNA or protein? What is actually in those genes that they code for?
Mm,
Are there some specific regions?
yes, ma'am. These are exons, the code for protein.
Okay, and are there only exons within the gene?
Ma'am,
Introns also.
there are introns and exons, but during splicing, the exons got combined. They get linked with each other and form the protein.
So one thing that they have exons, and exons in different combinations because of splicing regions, they can have alternative type of mRNAs. Is that you mean? And
Yes, ma'am.
and
Ma'am,
Yeah,
I
one
think there are
second.
exons and introns. When introns get terminated and the exons get joined with each other and they form an mRNA, which after that code for protein and translation takes place after that.
okay, so if I know the property of a gene. For example, I took a simple gene. Let's say there is one gene which is ribosomal RNA. Now, ribosomal RNA can be of different types, but there is one coding portion which is actually, or which is actually, we there is a stretch of nucleotides for which we call this, it is a 5.8s RNA. Or there can be a gene which has a conserved domain region. I expect you to know that a gene also has a conserved domain when it translates or transcribes first into mRNA and into a protein. So this protein eventually has a conserved domain, and for this conserved domain we call that this is the particular gene A. For example, if it has a mad box type of a domain region, or it can be a homeo box, it can be a yabi domain. So gene has that property for which it is known for that particular gene. And if I would ask what genome has properties, can you tell me the properties of a genome that distinguishes one genome from the other genome? If I say that A is the one plant genome and there is a P plant genome, so what properties makes them two different genomes? Because at the end this is a stretch of nucleotides only, but there would be some features which make them or which distinguish them into A and B. So is there any answer to this question? Can you tell me few properties of a genome?
They will express different proteins depending on their conditions.
I'm
Okay,
the sequence of nucleotides.
okay. And
There could be a jumping genes to distinguish between the two genomes.
okay, these are transposons.
Yes.
And
Length of nucleotide set.
so genome is defined by the presence of transposons. Okay. And
Ma'am, there are proteins with them specific.
uh, I think there is approximately 98 or 99% of gene which is common to all the genomes, and there is hardly a 1%, maybe less than 1%,
Ma'am,
which
actually,
distinguishes them. Hmm. Yes,
ma'am,
yes.
ma'am,
Hmm.
ma'am, there may be difference in splicing.
Okay. Or and
The enzymes involved can be different also.
and
Length of nucleotide.
so basically we do very little on genes and genomes. So for you, I repeat it. We know that a gene has a 5' end, which we called is a promoter region. Have you heard of this word?
Yes, ma'am.
Yes, ma'am.
So promoter
Yes,
is
ma'am.
the promoter is the region which suggest or which tells us that what kind of a domain or what kind of the other factors would come and join to it. So there is a specific promoter for the gene. There are further enhancers and silencers added to the gene, and gene has further their exon and introns. And when gene translates or transcribes first and translates into protein, and it has a conserved domain. So to distinguish between those genes, you need to know about the conserved domains. One, because if you know that this is the domain or conserved domain region of the gene, then only you will be able to distinguish or find out or fish out that particular gene from the database. No, if you don't know this myoglobin gene has this and this special domain, and if and you have no idea about this domain region, and if you will start searching on the databases, so what would you think that you would find? You will get false results. There will further be all results will be negative. So if you don't know the properties of a gene, you cannot use those bioinformatic tools for your research. So therefore, it is required that your fundamentals on the biology should be very crystal clear to you. Then only you can use those databases. And for genomes, I would say that there you may have heard of GC rich region,
Yes, ma'am.
which varies from genomes to genome. Animal have different composition of GC rich region, plants have different composition of GC rich region, and within plants there is also a distinguishable category. Other than this, you have microRNAs, you have small nuclear RNAs, you have a specific family of transcription factors, you have particular percentage of coding genes, you have also non-coding genes, you have satellite DNA, you have repeat regions in the genome. So unless you don't know the properties of a genome, how will you handle the genome first? So before anything you want to do in bioinformatics or you want to learn the bioinformatics, make sure that you know the genome and gene better. Because if you are thorough with that gene, that yes, it has a specific stretch of nucleotides which makes it distinguishable or which distinguishes those particular gene from the rest of the genes, then only you will be able to fish out that particular gene, otherwise not. Therefore most of the bioinformatic tools we see or we handle, we usually don't think so, and there at that step only we do various different kinds of mistakes. Fine. So I've told you about the genome. So genome has many features. There is a different composition of transposons. You may have heard of copia type transposons or LTR transposons, or there are microsatellites which are specific to it. There can be a different centromeres, or there can be a specific size of a genome which distinguishes from other. There are other ploidy levels. So you know all those things before doing anything. Okay. So this is the first database or very early database, and you all, I expect you all students that you have heard of this database, which is National Center for Biotechnology Information, now which we called it also as NCBI. So I've just googled this database. Now this database holds all or have all kind of biological information. You search any system, any system from viruses to fungi to alga to plants to animals to animal kingdom also, you will find here the sequences within NCBI. So this is the uh, early database, or I could say that this was first developed database. So when you want to know that, for example, you are working on a particular system, or you like one or two the other plant. So if you want to know about the plant on the database on NCBI especially, so you have to make sure that you type that botanical name of the plant perfectly. For example, if I'll show here, can you see in this box genome? So this, if you will click this arrow, there are many options you will see, because this is a snapshot, or I've taken this picture. I cannot use this site. It's just to show you that if you type where it is written a genome, so if you will type here, there are many options available for this. You can search for a genome, you can search for a gene, you can search for proteins, you can search for splice variants. There are different options available for it. For example, now I have typed here a Solanum lycopersicum, and if you make a mistake while entering the name of the plant here, you will not get the this result, because it is very specific. If you write a wrong name, or if you make a single mistake of a single alphabet, you may not get the result. So while handling these tools, you should know that whatever you type, that should be very uh, uh, that should not have any mistake. For example, I want to know about the Solanum lycopersicum genome here. So I typed in the genome category and selected this genome. And here I have typed the Solanum lycopersicum here. So there comes an information only on Solanum lycopersicum here. So these are all things that we can get from this site. For example, so where this plant actually fall across the plant kingdom, that the lineage, it belongs to the classes and different orders, family, different clades have been given here. And it also gives an information that here you will see that it has written there is a sequence data like genome assemblies or sequence reads. Statistics may require there is a protein count of this much, GC content of this much, and there is a this much megabase per genome. So from this typing this name and genome, you come to know that what kind of a data it has, how much is the genome size, how much is the GC percent, and whether it is a completely sequenced genome, or whether it is a draft sequence genome. Because there it is written in draft sequence genome that you have a scaffolds. So scaffold is a word which has been used to categorize or to demarcate those genomes which are not fully sequenced yet, up to the chromosome level. Complete sequence genomes, you will find that all the genes and proteins and whatever is in the genome, they are all marked on the chromosomes. So from typing right plant name or right system, you can come to know this much information from this site. Now, if I want to find a gene, I would go to this category where I have selected previously the genome, where their gene is also mentioned. So I will select this category and will type the name of a gene. For example, I have maybe due to this panel you might not be able to see, but I have written a specific name of a gene. If I make a mistake while entering its name, results would not come to it. So one should not think whether it is not in the database. So you please make ensure that you have corrected the entry correctly here. So what I did, I type a gene name here. So this kind of information came here. So these are different type of genes you can see. So this is, for example, I typed the AFO gene on the top. So this kind of a description that what it is known for in a specific plant. For example, an Arabidopsis plant, it has a specific this description. So it is named by plant specific transcription factor YABI family protein, and there you can see that this is a chromosome number 2. So this gene is on chromosome number 2, and this is the chromosomal coordinates. So this is a chromosomal position of this gene on the chromosome. So if suppose I gave you a name of a gene, fine, and I told you that you please search this and give me all the details of this gene. So so what I do that you find that these are the genomic coordinates of this gene, and I want you to use those coordinates and extract all the sequences available within this. So how will you go about it? Just think that I have given you specific coordinates, genomic coordinates of a specific stretch of nucleotide, and I told you that you please pull out those sequences. So how would you go?
Excuse me, ma'am. Hello. Excuse me, ma'am.
Yes.
Ma'am, could you please repeat from the given set of genes, then how we have to go further?
Okay, from this slide only, na?
Yes, ma'am.
So suppose in your mind there is gene A. You want to know about that gene A fully. You want all the information on gene A. So what I told you that you should write the name of the gene correctly. Then only information will appear on the screen, otherwise not. This is the first mistake that students do always. They don't write or they don't enter the name correctly, and then they think of, okay, this is not available in the database. So you have to be sure the name of the gene, the name of the plant you want to search for, you have entered it correctly. And for example, I have just to show you here, I have typed AFO gene on this panel, which is uh, this was the panel, and I have typed it here. Just because of this dialog box, I could not show you. So now so on the screen different type of genes have appeared. So what I told you that this is the gene name, this is a specific ID of that gene. This gene has a description. It is saying that this gene belongs to a family of transcription factor which has a YABI family protein. It means you can say that the gene I am searching for has a conserved domain, or is a transcription factor actually, and it is located on chromosome 2, and which is on a which has a genomic coordinate of this position. So this is the on chromosome 2. If I'll put these coordinate onto chromosome 2, I will eventually find this gene. This is I am trying to say you, and complement because it is in a reverse orientation. It is aligned from 3' to 5' direction. And these are the names for which this gene is known for. For example, AFO is also known as AT2G4Y190 abnormal. So these are the other names of this gene. My question was to you, if I am telling you I want the sequence stretch of nucleotides from this position to this position, how would you go about it? How would you retrieve those sequences? Clicking by
repeat the question.
You still didn't understand?
Ma'am, the question.
Okay. I'm telling you, I want you to extract the sequences of this particular gene. What will you do on the database? Would you click here?
And nucleotides.
Sorry?
The click on gene name, ma'am.
Sorry?
Ma'am, in the option above where we have chosen gene, we can select nucleotide so that we get the sequences.
So you will click here. You're saying this, or you will click here. Okay, you are saying you will click here and you will get the sequence.
Yes, ma'am.
Okay.
No, ma'am, I'm talking about where you have chosen that the search option where we've chosen gene, we can choose it as nucleotide.
Okay, I still didn't get your answer. Okay. So now what I did, that I clicked here and marked here, and this screen appeared to me. So this was like AFO plant transcription factor because I clicked this for gene. Now the information, it came to me that this gene is a protein coding type. This locus tag is an accession number. You can say this is 82G45190, and this is a gene specific locus. So when we code for, and when we want to code for particular gene, we use this tag. So if somebody reads, somebody have mentioned this kind of where 80 is for Arabidopsis thaliana, 2G is for chromosome number second, and this is the number of the gene. So this means that this is referring to this AFO gene of Arabidopsis thaliana. So it is also written, the gene is also written as this. So this is the information which has been given, that this is the function of this gene. This gene is also known as these by these names also. This is the classification of the plant that we have chosen. And next is that there comes a genomic coordinates. Now in the next you will see that location is chromosome 2. The gene this is a chromosomal position that has been given because it was a completely sequenced genome. अगर ये draft sequence genome होता, there you would not find the chromosome position. Now this gene has 7 exons, and this is the genomic coordinate, and this is how a gene is arranged in a on the chromosome. So this is a AFO gene. There it was written it is a complement gene, means it is in a reverse direction. Usually we always refer it to 5 prime to 3 prime end, and there it is in opposite to this 5 prime to 3 prime. So therefore it is showing opposite arrow. These are the other genes which are located or which are present next to AFO. So these can be said as that these are the upstream genes of AFO, and these can be the downstream genes of AFO. So this stretch is complete AFO. Now, if you see here, so this is the AFO gene where it is showing a 7 exon region. And if you want to obtain the genomic sequence or nucleotide sequence of this gene, you will find, or you will click the FASTA option here. So most of the databases most of the databases have information or nucleotide information in the form of FASTA. In my next few slides, I will show you that what exactly is a FASTA format, and what and how it is written. So any stretch of, and most of the databases that we use today, they take, or they take query sequences, or they whatever they we mine sequences from there, they all are in FASTA format. So one thing you just put in your mind, if you are entering, or you want to search for a gene, you want to retrieve a gene, or you want to use it as your query and find out the other genes, your gene should be in a FASTA format. Fine. So if this is the genomic coordinate, and these are showing the 7 exons region, these green boxes are the exon regions, and these are the, again, the this much, this is 19 million to this, and this gene is of 3286 nucleotide. From this panel, when you enter it on NCBI, you came to know that this gene is of 3286 nucleotide length. So overall information. So information on a gene you can extract from here, and this much information you get to know from this site. So because I clicked further here. So when I clicked this, these exon region showing here, so what comes is this screen. Now it is showing 4 different colors you can see here. So this is the showing the nm, there is some number with nm as a prefix, and this is np. So it is showing that gene which has 7 exons, it is giving 2 kind of mRNAs, nm for this mRNA, np is for the protein. So 2 types of mRNA are there, and therefore 2 types of protein are produced from this kind of a gene. So if you will click here, you will come to know the mRNA information, and if you will click NP, so you will come to know the protein information of this gene. So now from up till here, I hope that you have understood that how can you go about a gene, how can you enter your gene of interest into the box and retrieve its all information from NCBI. I hope it's clear to you all.
Yes, ma'am.
Only one student.
Ma'am, I have one doubt, ma'am.
Okay.
Ma'am, why it is mentioned the gene as only protein coding? Because all genes are protein coding, right, ma'am? What are other types? If I
Okay, let me repeat your question. You want to say why it is written a protein coding gene?
Yes, ma'am. Because all genes are protein coding only, right, ma'am? What are other types? I'm
Okay,
asking.
okay. I actually this can be more understood when you do hands-on like training onto it. Okay, for example, I give you some mitochondrial gene. For example, MATK. MATK is a gene. Okay, and this is present in mitochondria. Fine. I want you to enter MATK Arabidopsis thaliana on this NCBI site. Fine. So you will not get this kind of information because this is not protein coding gene. My aim is to make you aware on this site how you should use it and what extent of information you can retrieve from this site, because once you will have this information, then can you go further on any other database.
Yes, ma'am. So that gene
Okay.
could also be not coding, right,
It
ma'am?
depends on your gene. Now which gene you are entering, what that kind of information will appear on the screen. And these databases are developed so it makes your life easy, that you do. For example, okay, I have another question. If suppose this database is not there, you have a name that you want to know about one particular type of a gene from a system. You want to know that whether this plant has a type of a gene or not. आप क्या करते हैं?
We have to sequence and do gene annotation
How would you sequence it?
by NGS.
So first, what would you do? You will isolate a DNA of a plant.
Yes, ma'am.
Okay, the 2 days will go in isolating the DNA. Fine. So next step would be, because you are interested in one particular gene, you have to make primers for that particular gene. You will make primers for it. So the next 7 days will waste for primer making. Fine. So one day you will run PCR on it. Fine. And then in the next month you will give for sequencing, and about after 2 months you will get the sequence and you will come to know, okay, this has this kind of a gene. When database is giving you this kind of information in just 1 minute, why to go a long way na?
Yes, ma'am.
when database is there, so much of information is there, everything is there. You just have to use right kind of a tool for it. Okay, I expect that this is a new field. We do not know with what is the application of this field. This I will definitely talk about in the later, because I want you to be aware of it, because when you start using this tool, then only you can go to some extent and resolve your questions. So by wasting 2 months, when I have an information, there is a database available, there is a plant specific database available to you, and I am giving you an exercise that I want to know about a gene. So in 1 or 2 minutes, and maximum 5 minutes, if you know the tool better, so you will get all the information. Yeah, that this is a gene, it has a splice variant, it is located on chromosome this, it has a promoter or sequence this. So ये इससे कितना easy हो गया ना. Now you can go to the next step in a 1 day or a 2.
Yes, ma'am. But I got doubt whether the gene could be non-coding.
So if I search for non-coding gene, they will also appear on the screen. It is just to because I have been given a limited time, so I am so I have put it for one example here.
Yes, ma'am. Thank you, ma'am.
Okay. Any other question, that why to use databases, or why are we using it?
Excuse me, ma'am. Hello.
Yes, yes. Yeah, I can
Excuse
hear
me,
you.
ma'am. Ma'am,
Yes.
I wanted to ask that why is this gene from 3 prime end to 5 prime end? Like we always read the genes from 5 prime to 3
Okay,
prime end. What's the
you know
concept
that
behind it?
you know that the DNA is double-stranded.
Yes, ma'am.
Watson and Crick model.
Yes, ma'am.
So both the strands are in a opposite direction. One is in 5 prime to 3 prime, and the other opposite is 3 prime to 5 prime.
Yes, ma'am. So we are talking about the antisense strand over here.
Yes.
Okay,
What
ma'am.
matter?
Okay. This gene is present on the
Yes, gene is
present
present
on
on
the
the
antisense
opposite
strand.
strand. That's it. So its promoter would be
Okay,
on
ma'am.
different direction.
Okay, okay. I
But
get
this
it.
is a good question. At least you thought for it. Okay, why is it an opposite? Why it is a complement? I take it as a good question. Any other questions before I go ahead? No more questions. Should I go ahead?
Nisha, there are 3 questions on the chat window. Should I read them out or should we take it at the end?
Ma'am, you please read it, because it is difficult for me to handle so many things.
Rashi is asking what is FASTA format. Is there in the image? Is it there? What you
No, it is there. It's the next few slides, so I'll definitely show here.
And she's asking what is gene annotation?
Okay.
And how are chromosomal coordinates measured?
Okay. How are chromosomal coordinate?
Measured.
Coordinate measured?
Yes. How are chromosomal coordinates measured? I request Rashi if you can clarify to ma'am. You unmute yourself and please clarify.
Ma'am, my question is how did they know, like how did they measure the chromosomal coordinates on the DNA?
Okay, and your first question was that gene annotation. That what is gene annotation?
Yes, ma'am.
For example, there is a plant. देखो, one always put a standard genome. For example, Arabidopsis thaliana was first fully sequenced genome among the plants. We know each and every bit of it. Yes, this is the coding region, this is the another region. Now I go to the other plant. For example, I go to tomato plant. Now I isolate a gene from there. Now this gene which I took out I just know that this is a stretch of those nucleotide sequence. Now this stretch of nucleotide sequence, whether it is a coding gene, it has some conserved domain or not, it has a some exon intron structure or not, and how is it similar to the reference genome. So when I match my gene with the reference genome, I came to know this is 90 to 100% match with the reference A theliana genome, and in A theliana it is a type of a protein, and therefore I also called my sequence, or which I am working on, I have put it in a I have put it as that this is also a type of a protein in tomato species. So this is what, when you give a name to a gene on the basis of using some other reference genome which is completely sequenced and completely analyzed, where we know there that it has this gene. So when we match it, then we say that yes, I annotated it's this gene because this gene has no identity in the database, and when I use different tools to look out that this gene is much similar to the a gene in Arabidopsis. So I have given this a type of a gene in tomato plant, and this is what is called gene annotation.
Thank you, ma'am.
Still, you understood some of it or
Yes,
not?
ma'am. Yes, ma'am. Yes,
Oh,
understood, ma'am.
okay. And then your second question was that how do we give this? How do we know the exact numbers of this gene?
Yes, ma'am. The coordinates.
Okay, this question would be very clear when I will discuss the sequencing, that how genome is sequenced. So when you know that the genome has been sequenced and it has this much base pair and they are like this, then you will come to know, okay, this then you will be clear with this question. If I'll speak now, you will not be able to understand. So I think you
All
wait
right,
for at
ma'am.
least few minutes, no?
Yes, ma'am. I'm so
Okay.
sorry.
Okay. Okay.
So Nisha, we can take the
Uh,
question answer round at the end so that your
okay.
flow is
Okay.
not disturbed.
Okay. So from this slide, we came to know that this gene has to actually producing two kind of mRNAs, means that there is an alternative splicing here. It just okay. For example, now from the previous slide, uh, I told you that these are the genomic coordinates of a gene. So I have given you an exercise. You please give me the promoter sequences of this gene. Fine. Suppose this is a coordinate which is there. We know that this gene has a this genomic coordinate. For example, it was written. These are the genomic coordinates, and I want you to isolate promoter sequences of this gene. Means promoter sequences, meaning that if this is the 5 prime end of the gene, from here, from this base pair, if first nucleotide base pair starts, so promoter region will be will be a 1.5 kb away from this gene. So, and I want you to isolate sequencing sequences for me. So how will you go about it? Now you see that, for example, this is a another tool actually which we called as a genome browser tool. Genome browser tool means if you want any kind of sequence, you know that these are the coordinates, and you want to extract 200 base pairs upstream of this gene or 200 base pairs downstream of this gene. So uh, instead of counting base pairs, there is a tool which we called as genome browser tool. So every site you will use or you will see has written on that site that there is a genome browser tool. Now if this on this site, now you will see here, this is a chromosome 5 they mentioned here, and these are the genomic coordinates. If for example, now this is a suppose base gene located at 5 lakh 25 52 lakh 53,001 base pair. If I delete from here 100 and go upstream. So what I'll do, if suppose you want to take out sequences 1500 base pairs upstream of your gene sequence. So what will you do? You minus 1500 base pair from this. So 5253001 minus 1500 base pair. So the number that would appear on your calculator, if you'll put here, you will get a sequence with you will get a sequence upstream of it. For example, now suppose this is the region which I have minus here and put it here. So when I'll go to the reference sequence here, so this is the visible region. Now this is the region, for example 5253, this is the same number which is mentioned here. So if I'll change this number 1500 minus here, and we'll view it, so I will get the exact sequence. For example, I will get the gene sequence as well as 1500 base pair upstream sequence also. So if you will do it manually, you will take many days to do it, because on Word or any document you will do, it will take very much time. It will take so much of your time. Now, so if you know that there is a genome browser tool, you can put any number here you want to isolate, and your sequence will be retrieved very easily, because once you click this view button, you will be able to I have not shown here, sorry. So your all nucleotide sequence would appear. So this is the another tool. And second important thing is that suppose this is your gene of interest, and you want to know that which are the upstream genes of your gene of interest and what are the downstream genes. So you see that these are the genes which are actually upstream present on your gene of interest, and these are the downstream genes which are present, and they all with the annotated protein. So you can come from just in a minute, you can come to know that yes, these are the upstream genes, these are the downstream genes, these are the specific locus number of your gene, and this is on 5 chromosome. And if you want a promoter sequence for your gene or enhancer sequence, you just delete how much upstream you want to go. So you change the number here if you want to go upstream, and here if you want to go downstream, you will get your desired stretch of nucleotide in a single minute. Okay. So one question was that what is FASTA format? So if before this stretch of nucleotides, you put a greater sign and mention it, this is referring to, because this is a sequence from A theliana, and this is a protein sequence, because these are the what are actually these? How can you say this is a protein sequence?
Single code of amino acids.
Repeat.
Single letter code of amino acids.
No, these are amino acid
Yes, sir, it's
initials.
present, ma'am.
These
Yes,
are
ma'am.
amino acid initials, and therefore we are calling it as an protein sequence. And this is a nucleotide sequence. It can be a mRNA sequence. It can be a gene sequence. So it is stretch of a nucleotides. So FASTA format is actually a greater sign followed by your sequence. If you'll not mention your plant name, then also databases or any bioinformatic tool would accept this as a FASTA format. But you always have to remember that there should be this sign before the sequence. Then only it will take and give you results. If you will just paste sequence as it is without this greater sign, you will not find any result. They will show you some errors, and that work won't happen. Okay. And this is the protein sequence, because This is the all initials of different kind of amino acids, and therefore be called as a amino protein FASTA format of protein sequence. So this is basically a FASTA format. Most of the softwares uses this kind of a tool only. Okay, use this as a format for for you can enter data like this only. Okay. Any other question?
Ma'am, these are
Hmm.
ma'am, these are amino acid sequences. The one which is
So this
below.
is for like M for methionine. So the protein sequence is written like this. So every initial specifies an amino acid. No,
Ma'am, what about F and K?
that is F. F for
F is phenylalanine.
what?
F is for phenylalanine.
Phenylalanine. Yes, phenylalanine.
But that
And
starts with P, right?
Yes, but some amino acids have different
you
names.
Yes.
have to
All
see,
right.
but
All
this
right.
is definitely a protein sequence. Because I really don't remember which is actually a signature for which is actually a letter for each kind of amino acid.
All right.
P is for
Thank
protein. That's used for
Uh,
protein.
hmm,
That is
hmm.
why phenylalanine is given F code.
Okay. Hmm. Thank you, Divya ma'am. Okay. Now, these are some of the other bioinformatic tools you use. So if your which is we called as a BLAST, this is a basic local alignment search tool. This is the full form of a BLAST. So now BLAST tool actually based on your similarity index. For example, if you put a sequence, for example ABCD, and database has a sequence similar to your sequence, your query sequence, that on this similarity basis, that how many nucleotides are similar in a stretch, they are in a stretch of sequence, so they calculate as a percentage similarity, and there you see the results. For example, now these are different kind of blast results, and they have all have a different meaning. So when your sequence, when you have some gene of interest, I would say that is your query sequence, and what is present in database, that is a subject sequence. So when your query sequence is a stretch of nucleotides and you blast it with the database which is also have a stretch of nucleotides, means your sequence as nucleotide a stretch of ATGC, so it would find similarity from the database with the nucleotide stretches only. So this is therefore this is known as nucleotide blast. So when your translated nucleotide, for example this is your suppose this is your mRNA sequences, and you want to know that which kind of a protein it is, so you will use this blastx tool. Similarly, if you have a protein sequence and you want to find out on the database that how what is it exactly matching with some mRNA of on the database, so you use T blast m. And protein blast, when your query sequence is a protein and you are searching against the protein database. Did you get it?
Ma'am, could you please repeat this once again?
Okay. Okay. Um, Suppose you have a stretch of nucleotides, 20 base pairs you have. Fine. You want to know these 20 base pairs. Your 20 pairs would be referred as query sequence. So database would take it as your query. So this is your query sequence. So it would appear on the screen as query. So sequences already present within the database would be uh, your subject sequence. So they would appear on the screen as subject sequence. Now, when you use your query sequence in the form of nucleotide, a stretch of nucleotide, and blast it with the same nucleotide database, you called it as a nblast. When you use protein sequence as a query and blast it with the database which has protein sequences, so this is known as protein blast. So protein against the protein database, nucleotide stretch of nucleotide against the nucleotide stretch. And when you have your mRNA sequences, you want to know the proteins that it codes for, or that it shows similarity with. So you will use blastx tool. And similarly, you have protein as a query sequence and you want to blast it against, you want to find that which mRNA is there on the database which shows similarity to your protein, then you will say that this is tblastin tool you will use. Now, for example, when I click here, this is nucleotide blast, and for this screen you have to type uh, NCBI blast, because this is the tool which is available on NCBI, or this is a tool from NCBI only. So if you type on a Google NCBI blast, this screen will appear, and you should have some query sequence with you on. If you click this nucleotide blast, now what would this screen will appear? Now what does it mean? It is saying that enter query sequence. Query sequence are those sequences which you are using for which you have. You want to know your sequence कि ये किसके साथ ज्यादा similarity दिखा रही है, किसके साथ नहीं, ठीक है? So if you use yours, so you will put sequence in this white box here. Now you can use accession number also. Suppose you have seen that in the database your gene, I have already mentioned that there is some locus number. If you put this locus number which also uh, is your gene, which is your gene, but and you type accession number here, or you can use FASTA format here. You have sequence, you put a greater sign here and enter the sequence. Now we usually set database as non-redundant database. So most of the time only this kind of non-redundant database you will use this option only. And if you want to use your query sequence, you want to blast your query sequence with a specific organism. For example, you have isolated a sequence, or you have a sequence, you know, from this is a potato plant, fine, and you want to know which gene is actually in this potato plant. So I will put here enter organism's name mein, this is Solanum tuberosum. So suppose you will put here in the organism section, you will put here Solanum tuberosum. Eventually most of the taxa with Solanum tuberosum will appear and you will select it, fine. And if you don't want to do it, you can retain it as it is. So this is the only option you have to choose. You have to enter your sequence with greater sign here. You have to select always non-redundant database. And if you want to do blast against the complete database, you need not to enter anything. If you want to blast with a specific organism, you have to mention here. If a database has that has that organism or has that plant, it will take it, and if it is not there, it will not show you, and so you cannot select it. So you have to blast it against the complete database. So the next thing which will appear, this is a program selection. So this is third thing which you have to somehow enter, because these things cannot be this needs to be filled. You cannot just uh, uh, escape all these things. Now third thing is program selection. Now it is saying that it is highly similar sequences, there are more dissimilar sequences, and these are somewhat similar sequences. So most of the time what we do, we always select somewhat similar sequences, because it what does it mean? That stretch of nucleotide you have, it has a higher probability that at 70% of that stretch would definitely be identical to the sequences present on the database. Because sometime what happened when you select this highly similar sequence option, no, so what happens, 100% of sequence, if you want exactly identical sequence on the database, so you have very less chances, or sometime it shows you the error. So you should start with this somewhat similar sequences, so there are high chances that your query sequence would definitely this take this blast option and will give you results. So try to select somewhat similar sequences, so it will give you result in a uh, few minutes. So once you do this, you click this blast tool. So you click this blast option and results would appear. So this is how I put it sequence here, which I was showing you, that it has a greater sign and followed by the sequence. So I have to always you have to put sequence in a faster format here, or if you know the accession number you can also use, but that also with the greater sign. And see, second option with that I have selected is nucleotide non-redundant database, that you always have to select. And this is how this results come. For example, I used one of my query and blasted in NCBI. So results would appear like this. Now, how would you interpret these results? So uh, actually these are, I don't know whether it is these options are appearing on your screen or not. Is it appearing? Can you
Yes, ma'am.
read these
Yes,
boxes?
ma'am.
Hmm?
Yes, ma'am.
Okay, so I think this must be your coverage. What is this option exactly? Can you read it for me?
Accession.
Accession.
Huh? And this option is query coverage.
Accession, madam. It's
I think this would be the percentage similarity, and this would be your query coverage, and this would be E value. Am I right?
Yeah, we can see till exertion.
Okay. Okay. Now, how would you interpret your results? Now you see. These are the different sequences which were present on the database. So when you blast it with on the database your query sequence, so it showed similarity. It means that there is 100% similarity of your query sequence with A theliana plant, and this is the specific gene. So, and there are other so this is also an Arabidopsis thaliana same clone, and this is a complete sequence, and this is showing 99.78% similarity. Similarly, there are other genes also which are showing um, less than uh, this Arabidopsis thaliana is showing 100% similarity here. 80s may in these some of these brassica species are showing 80% similarity, and this genome assembly has been sharing this much percentage. And now there are also some sequences which are showing 100% or 90% similarity there. So can you tell me which is the most reliable sequence that your query
100%.
So can I select, should I select all sequences 100 with 100% as my subject sequence? So can I say that this is Arabidopsis thaliana clone sequence CRC, and this is this so all these are representing with 100%. So should I consider all these as my true my actual sequences? Because I don't know my query sequence, what it was coding for, I don't know that. But when I blasted on a database, these results came. So you are saying if it is 100%, I should say that this is genome assembly chromosome 1, or this is this Yabi family, or if it is showing 100% or 96% here, I should say this is Brassica repa. It could be the 3 or 4 options. Any other guesses?
It should have maximum score and less C value.
Maximum.
Maximum score and less C value.
So which is showing you the maximum score here?
First one, ma'am.
And and that genome assembly has much higher number, 30272,351. It has much higher number. Why not this? E value has 0 for all. Any other guesses? I think 2 or 3 students are just listening.
Ma'am, is there any relation of this with query coverage?
That I want you to think now. I told you that I put my query sequence on the database. I found these results. There are 100% similarity with this sequence on the database. There is this much percent similarity with this much high number with Arabidopsis thaliana genome assembly chromosome 1. So which sequence I should refer is the actual sequence. So I should say my query sequence as assembly chromosome 1 or Yabi family or what? Because one person is saying maximum score, and listen, this is also a maximum score. 1131 से ज़्यादा तो 3928 दिख रहा है मुझे, and that too in Brassica. So one could say that it is it could be Brassica repa protein. So it must have isolated from Brassica species. Yeah, it is. You can not take it for granted that the first sequence which appeared first on the screen has a higher percentage of similarity. It may be on NCBI it is showing you this, or if you'll handle other databases, you have to be sure that which criteria should you use, that should be stringent, and the sequence you're actually referring to or annotating your gene should be true. Because if at this step if you do mistake, your whole analysis, your whole further analysis will be all waste. So it will all be wasted now. So initial steps should be, uh, you should know that which sequence you should choose, so that any downstream analysis. Hmm. Yeah, somebody's.
Ma'am, we can't select sequences with 100% similarity as all the sequence will be similar. Therefore, we won't be able to observe the conserved regions.
So in short, you are saying that most of the sequences which are showing 100% here, so they are actually not your query sequence. They are not identical to your query sequence. So I should forget that. Definitely my query doesn't belongs to this. It doesn't belong to this gene. So there should be some criteria now.
Ma'am, can you please tell what ACCLEN is?
Where it is?
Ma'am, the second last row.
Second last row.
Near to percentage identity.
Actually, this is not appearing on my screen. That's why I asked you to, tell. Okay, wait.
Ma'am, the percentage identity should be 90 to 95%.
Okay, and more than that is harmful for you, huh? It is harmful for your sequence. And remember one thing also. It is not necessary you know each and every bit of it, but rather I would say you know which should we consider it, which is the most important criteria, and based on that criteria you can filter many sequences. All words appeared on the screen need not to be इतना भी जरूरत नहीं होता कि सब चीज जाननी चाहिए. जो ढूंढना है ना वो पता होना चाहिए कि yeah, this should be the criteria. I should see these two factors, and based on it I should retrieve and isolate and these are my actual sequences. So I don't think that I don't think that anybody actually know what is the criteria to filter those sequences and filter or fish out your gene of interest within this database. I don't think so. So first most important criteria which you should always think of should be query coverage. Now it means that, for example, your query sequence was 50 base pair sequence. Fine. So all 50% base pair sequence exactly matches with this Arabidopsis thaliana plant transcription factor YABI family mRNA. But if the query coverage here is 87%, it means 30 to 45 base pairs of query sequence are exactly identical to Camelina sativa protein. So if you put a query sequence of 50 nucleotides, so all 50 nucleotides should be identical to the sequence present on database. For example, here the 50 base pairs I have entered as a query sequence, they all are identical to this protein. But here for this gene, here the query coverage is 51%. It means 25 base pairs of my query sequence are 100% identical to this protein. So you tell me, if my query coverage is 100% here, and here 25 base pairs are identical to this protein, and here all my 50 base pairs are identical to this protein, so which should I choose? Which should I rely on? Yeah, this is the exact sequence which I was searching for. This is the exact gene. Of course, all 50 base pairs are which show exact identical, they show exact homology or sequence identity to the sequence present on the database. Now result similarity index का तो यहाँ भी same था ना, 100% you are getting here also and you are here also. Now here 50 all your base pairs are identical to this, and here only 25 base pairs. So when my all base pairs are identical similar, they are blasting with the against the database which complete 50 base pairs. So I should rely on this, that this first sequence should be my subject sequence is my target. So my query is actually a Yabi family protein, and this is from A. thaliana plant only. So it means the query sequence which I have chosen as an entry here, it is nothing but a transcription factor from Arabidopsis thaliana plant. Now you tell me, यहाँ पर भी same case है. देखो, this is 100%. यहाँ पे भी 50 base pairs covers the sharing the 100% homology or sequence identity. तो फिर ये क्यों नहीं है? Did you get what I am trying to say you?
Ma'am, would you mind repeating it once?
Oh my. And other questions? Okay, I'll repeat it, but before is it didn't you get?
Excuse me, ma'am. Ma'am, when the query coverage is 100% and when it's 51%, we can identify like which we have to select. But when the query coverage is 100% in both the cases, like you are telling right now, na the first one and then how can we be able to identify which one we have to work and which we identified?
This should be this would be definitely my next question, but up till here you understood that at least from 100% query coverage and from 50% query coverage, which should I choose? This much you understood.
Yes,
Yes,
ma'am.
ma'am.
And what about the others who are not saying neither yes, not no? Okay, suppose we should go further. Now there is a quest between this sequence. Now Arabidopsis thaliana plant specific this with 100% query coverage and 100% similarity, and second is that Arabidopsis genome assembly chromosome 1 with 100% query coverage and 100% similarity. Now for example, if this is chromosome 1 genomic assembly, for example, this sequence on the database, you think it is of 500 base pairs. Fine. You just think that, for example, this subject sequence is of 500 base pairs, and this sequence is only of 50 base pairs, and with 50 base pairs my query exactly shows sequence identity with this 50. Now in this 500 base pairs there would be 50 identical nucleotide base pairs which exactly matches with my query. So there can be a case where more than subject when there may be a stretch of nucleotide sequence would be larger in length, but that doesn't mean uh, कि होगा for it, what should I say? Okay, now for example, uh, suppose this is a 500 stretch of nucleotide, and from suppose this 50 base pair nucleotide lies from 1 to 50 and 56 to 50 500 base pair, there is no match between this. But there can be a stretch of nucleotides ना which should be, ऐसा भी हो सकता है कि एक कंस्ट्रक्ट का साइज न्यूक्लियोटाइड का साइज 500 बेस पेयर का हो, लेकिन उसमें 50 बेस पेयर, क्योंकि मेरा क्वेरी भी तो छोटा है ना, इट इज 50 बेस पेयर्स ओनली, और 50 बेस पेयर्स मेरे इस जीनोम असेंबली में ब्लास्ट हो गए, लेकिन इस क्रोमोसोम असेंबली में एक ही जीन नहीं बल्कि देयर वुड बी मेनी जीन्स होंगे, but here it is a one gene. So from here I cannot say that this is what kind of a gene. It is a protein coding gene. It is just giving me a location. So if you read Arabidopsis thaliana genome assembly chromosome 1, did it get is it giving me any information about my gene? Because it is long stretch of nucleotides and it is not annotated, whereas this gene is completely annotated, and I could say that this my query belongs to this protein. It all and what I can say further, that this sequence must be on chromosome 1. This can I deduce from here. So if I would say that if one sequence with exactly similar result, 100% similarity, 100% query coverage, so if it is annotated with protein, I would rather recommend this, because it is giving you an exact protein type your query should have. So your query is actually a CRC protein, rather than, because it is not annotated, na, it is just written it is a chromosome 1 genome assembly. It is not a gene, it is just a stretch of nucleotides present on the database. Any other question, or you didn't get it? Am I audible?
Yes, yes, you're audible.
Okay, so there is no question on this. Everybody have understood it that which gene should I choose? We choose for analysis now. So are they know what criteria should we choose for delineating our gene of interest? Okay, I'm hoping that they know, that they have understood. Okay. Now, for example, there is another tool. We called it is a spline tool. Now, this is also a tool from NCBI. I give you a gene sequence which is of suppose 100 base pairs, and I give also you a mRNA sequence of that particular gene which is of 500 base pairs. Now I want you to tell me how many exons that gene has, how many introns that gene has, and where is the exact location. So how would you go about it? Did you understand my question?
By transcription.
From transcript, for example, okay, you have gene A with you. Okay, you have some sequence, genomic sequence of gene A. I've given you that this is a yes, 100 base pair stretch of nucleotides of genomic DNA. I am giving you transcribed sequence of that genomic DNA, cDNA. I want you to tell me how many exons that gene has, how many introns, and where are its exact positions on the gene.
Ma'am, whatever found in cDNA is what are missing or introns?
You know that there is a specific conserved sites for intron starting and intron ending positions. From where intron starts and from where intron ends. There is, I think, as far as I remember, there is some GA site where splicing occurs and mRNA forms. So from fundamentals, we know that there are some genes, intron position starts from here, so we could hint, we could take, these hint can help us. Okay, this can be the exon, this can be exon. my, I just want to put this question in simple words, that how can you understand, for example, you isolated a sequence but you do not know where are the exonic positions of that gene and where are the intronic positions of that gene, but you have genomic as well as cDNA with you. So how will you map those cDNA? What would you do? Your cDNA is your exonic regions. Okay, so what you will do, you will see similarity between your exonic regions of cDNA with the genomic regions. Now if you do it, you can do this manually. Now cDNA is what? Your expressed gene portion, so it is exon only.
Yes, ma'am.
Okay, so if cDNA is with you, genomic DNA is with you, you see that okay, this is a stretch which is similar to genomic, you will say this could be the exon. Similarly, because cDNA is without intron, so if you will see the identity or you will match both the sequences, you can do it manually, but this would definitely take a time. So what this tool does, this is a spline tool. You have to just type NCBI spline, SPLIGN. What you do, this appears. For first, it appears as this. It appears this on the screen. You have to go online because all tools are available. You just need to have your net connection. So when you click here online option, so this screen appears. So if a gene sequence you have and if a cDNA sequence you have, so you will put the FASTA sequence in FASTA format here. So what I do, uh, I'll put cDNA sequence here and genomic sequence here, and will say click this option align. Now what would happen, this would come to your screen. Now how much time it would take? Hardly less than a minute. Maybe 20 or 30 seconds. Now this gene has been mapped, different exon and intron positions they are showing. This yellow box says you about the number of exons present. For example, this gene has 1, 2, 3, 4, 5, 6, 7. 7 exons, and these are showing you introns. Here 1, 2, 3, 4, 5, 6, 7 is telling you that these are 7 exon. This gene has 7 exons, and this is the region where exon lies. So you can say this base pair to this base pair. So this is how you do when you have information on cDNA and your gene, but you don't want to do it manually. You can use this tool and it comes to you information that yes, this gene has a this much exon intron. देखो exon में लिखा है this much percentage identity है, overall coverage 100%. CDS in frame, primary transcript means mRNA which I have entered is of 114.5 base pair. There are hardly 18 mismatches. Mismatches means there can be some matches which are not identical, so they are called as mismatches. And these are the exons. These are the base pairs given. Minimum base pair exon is with 54. There is a large exon of 163 base pair, this which should be this only. And these are the different size of introns present within this gene. So in just a second or a few seconds, you can come to know this information. Sometime it happens that when you retrieve a sequence from the database, for example, you isolate a mRNA sequence from somewhere from database A and from genomic sequence from database B. So from this tool, you come to know whether they are actually the true mRNA and genomic DNA. So if it appears on the screen, it means that you are going in a right direction. It is a gene, and the mRNA which I entered is actually mRNA of this particular gene. Okay.
Ma'am, should we also consider the poly A sequence at the 3' end of our mRNA?
So this is showing you that this is the first exon. So it starts from, it should start from, this is your 781. So this is the stretch of nucleotide which is starting from here. So it must be in some 600 kuch. Because most of the mRNA sequences which are present on the databases, there are very less sequences they are with polyase. They are usually partial sequences are there in the database. So they also cover polyase. अगर होगा तो वो map हो जाएगा यहाँ पे. अगर A tailing होगी mRNA में, this tool would take care of it. यही पूछना चाहते थे ना?
Yes, ma'am, because poly A tailing will be added after transcription only.
So
right,
if your mRNA has a poly A tail, you need not to worry about it. You just enter your mRNA sequence and genomic sequence, and you will come to know that okay, this is this much with exons, and these are the positions, and this is the this much mismatches are there. So on basis of it, it will assure you that you are in a right direction and you have chosen the right kind of sequences. Because here it is written that overall this is percent similarity, coverage is 100%. So the base pair is from 1 to 1792. So all information was they have been given on this screen.
Yes, ma'am.
Okay, there was one question which was asked that how do you know whether this is an actual this number of base pair? How do you know that this is 13 million base pair? This gene is starting from exact this number or ending here. This was a question. So for those questions, I want to introduce or show you here that how the sequencing has been done. So this is a de novo sequencing. De novo sequencing. De novo what? Have you ever heard of de novo word?
Synthesizing from scratch.
From scratch. Madhab, you are somehow near but not exact answer. with reference to this question, मतलब this स्क्रैच से तो हम बहुत कुछ शुरू करते हैं.
Like from the beginning.
हाँ, बहुत सारी चीजें बिगनिंग से ही शुरू होती हैं. But here there is, when we were sequencing or when a genome has been sequenced and annotated, there is no reference genome. Reference genome, because if suppose this is the, okay, wait. So if there is no reference genome, because suppose this is a completely sequence, we are not we are doing from a beginning, for example. So these are different kind of a reads. So I have use different kinds of primers here and started to construct or started to synthesize it. So but I aligned it on the basis of these ending sequences here, but I didn't match it with the genome which is already sequenced and annotated. So I have no reference genome. When I don't have any reference genome to which I can see that I am going in a right direction, or that's why it is called as de novo sequencing. For example, now see, this is whole genome sequencing. There is already a genome which is available on the database, and there is another kind of another plant or a system has been started has been sequenced. So on the basis of using this as a reference position, I have been able to make scaffolds or align these sequences based on it. And this is the case, this where we will not say that this is a de novo sequencing, because I'm using here this as a reference genome for this specifically or especially this sequencing this plant genome. But when I don't have any reference genome with me, we called it as a de novo sequencing. So there is a probability that we might come with a false results, because sometimes contexts are not aligned the way they should be. So this is how sequencing has been done. And have you heard of dideoxy method or Sanger sequencing?
Yes, ma'am.
No, ma'am.
And any project which has been sequenced through this project by this method, Sanger sequencing?
Human Genome Project.
Okay, and what is the principle of Sanger's method? That was dideoxy method. So you, when earlier this was, we said as a Sanger sequencing, but these earlier method, when we sequence a genome, we go from one end to the other end very sequentially and very gradually. This is the this, and therefore it takes so much of time, because there we fragmented the DNA, but we go in one particular direction. So next generation sequencing may there are different platforms, and therefore these now the plant genomes or any genome like human genome sequence took almost 13 years. Fine, and this much years because it did by a long sequencing method where you have to have one complete end, then only you can start the second end. But the now there is a different platforms available. Their main principle of these second generation, or we say it a next generation sequencing, is the multiple parallel times of sequencing. So this is the only advantage over the first generation sequencing. So this is the kind of how we sequence it. For example, These are, for example, earlier we used to make back libraries. These are bacterial artificial chromosomal fragments. So these backs can be obtained from bacteria, and where we combine fragment our DNA samples and join with the back ends, and we sequence it and we align it in a way so that there are some overlapping ends which are similar in all, and this is how we make a contic. So these consensus sequences based on these all short reads, when we collected different kind of consensus, if we try to join it through read pair or gaps, and this is how a kind of a sequencing we do it. So we have to go this way. So this is a long way of standard method of sequencing. So the other method is next generation sequencing platform. So at your undergrad level, uh, I would not want that you know all the principles of all these platforms available, but you should know that the next generation sequencing